So today I'm not going to talk about this, but I will get to this. Uh, Genes rearranging from stem cells to become B cells, but I still, it takes me forever to understand these. Like at this age of my life, it's hard. So what I'm just going to talk to you about today is, and this is like where I work. So that's that. But today I'm just going to be walking you through the process of how you create macrophages. So, how to create macrophages, I guess. So the first step is after you get your mouse and you sacrifice it, usually with like CO2. You put it onto like a sacrifice board, and then you remove the legs, and that's the femur and the tibia. And those have the majority of the bone marrow, so you don't have to really worry about the arms. So you remove the tibia and the femur, and then you clean it off you clean all the muscle off of it, so it's just the bones, and you put it into PBS. Once it's in PBS, you go to your tissue culture room because it's more sterile, and you aspirate all the PBS out, and then you put ethanol in there, 70%, and you shake it around so it cleans it off. Aspirate again, and from there, you dump them out onto a tray, onto a cell culture plate, technically, on a tray. And from there, then you use a razor blade and you cut the ends off of each of the bone so that there's openings on each end. So like the bone cap is cut. So, and what you do then is you take more sterile PBS and you get a 10 milliliter syringe and a 25, 28, one of those two gauge needle, and you fill up your syringe with PBS, and then you hold the bone with your forceps, and you take the needle and you stick it into the opening that you cut with the razor blade, and you flush it out with PBS, and all of the bone marrow goes out onto a dish. But that bone marrow has like a bunch of other stuff in it as well. I believe erythrocytes. And from there, what you do with that, after you repeat that with all the bones, you have a nice mixture, and then you run it through a cell strainer, just a basic one on like a falcon tube. And when you run it through the cell strainer, you have like a nice homogenized liquid at the bottom. Then you spin that down, probably 500 times the force of gravity, and you spin it for 4 minutes, and it separates out the PBS, which was used to flush, and the actual bone marrow. Aspirate the bone marrow, dislodge the pellet that formed at the bottom. Then you're going to add ACK, lysis buffer, and you're going to mix that with DMEM, which is just a type of media, and that's going to lyse all the cells, or lyse some of them. And then you're going to leave that in there for a minute, and then add one additional volume of ACK. So you're going to add some amount of ACK for one mouse, and you're going to add an equal volume of DMEM. And then you're going to, after 1 minute of that mixing, then you're going to add another volume of DMEM. And then you centrifuge that, spin it down, and you have your cells. Then you plate, and you're good.